نبدأ اليوم بتسجيل المحاضرة الثانية حول الهرمون of pancreas. The endocrine portion of the pancreas represents 1 to 2% of its total weight, and it is consists of 1 to 2 million islet of Langerhans that are collection of at least 4 types of cell, which is called the alpha or the alpha cell, beta, delta, and the F cell. These cells secrete at least 4 types of hormones: glucagon, insulin, somatostatin, pancreatic polypeptide or PP, respectively. Insulin, regarding its history, in 1889, both von Behring and Mikowiecki demonstrate that pancreatectomy in experimental animal produced diabetes. The link between pancreatic islet and diabetes was suggested by D. Meyer in 1909 and by Sharpe Schaffer in 1917. However, it was Banting and Best who proved this association in 1921. These investigators used acid ethanol to extract from the pancreatic tissue an islet cell factor that had potent hypoglycemic activity. This factor was named insulin, and within a year, both bovine and porcine insulin was widely used for treatment of human diabetes mellitus. Insulin was the first protein that proved to have hormone bioactivity, was crystallized, was sequenced in 1951, was synthesized biochemically in 1964, was synthesized by recombinant DNA technology for the commercial use. The chemical structure of insulin is a polypeptide, consists of 2 chains, the alpha and the beta, that are connected by 2 interchain disulfide bridge that connect A7 to beta 7 and A20 to beta 90. A third intrachain disulfide bridge connect the residual 6 and 11 of a chain. The A and the beta chain contain 21 and 30 amino respectively in most species. Porcine and bovine insulin was the standard therapy for diabetes. Before human insulin was prepared by DNA technology, porcine insulin was different from a human insulin in substituting alanine instead of threonine at B30. However, bovine insulin was this substitution in addition to 2 other substitutions at A10 and A8. Synthesis and release. Insulin is the first synthesized in the pancreatic cell, islet, beta cell, as a prohormone with a molecular weight of 11,500 Dalton. This molecule is directed by the hydrophobic 23 amino acid leader sequence in the crestina of the endoplasmic reticulum and is then removed to make the 9,000 diameter or Dalton proinsulin molecule starting from the amino terminal. This molecule is the beta chain connected peptide, C peptide in the A chain. As you see in this diagram, the structure of the proinsulin indicated the cleavage site at which insulin and C-peptide are produced. This proinsulin molecule undergoes a series of site-specific peptide cleavage that result in the formation of the mature insulin and C-peptide in equimolar amount. Proinsulin molecule varies in its length between 78 to 86 amino acid due to the variation in the C-peptide. Proinsulin and insulin have the same solubility and isoelectric point, and both of them form the hexamer with zinc crystals, which are present in the eyelid in high concentration. However, proinsulin has only 5% of insulin bioactivity, and a small amount of its normally secreted into the circulation together with insulin when the release of the latter is stimulated and is larger amount in the pancreatic tumors. Proinsulin has a longer half-life than insulin, and it reacts with insulin antisera in the RIA overestimated insulin bioactivity. RIA, RIA, is the radioimmunoassay abbreviation that is a way to measure insulin using radioactivity. C-peptide molecule have no known biological activity and with no distinct antigenic property, and that's why its measurement can distinguish endogenous insulin from that given exogenously as a therapy. The human insulin gene was identified to be located on the short arm of chromosome number 11. Regulation of insulin secretion. The human pancreas secretes 40 to 50 units of insulin daily. A number of mediators are implicated in insulin release, including first, glucose. High plasma glucose concentration is the most important physiological regulator for insulin secretion. The threshold concentration of fasting plasma glucose for insulin release is 8 to 100 milligram per deciliter, while the maximum response is at fasting plasma glucose of 300 to 500. Two theories can explain the role of glucose. Glucose receptor at cell surface of beta cell, rate of metabolite influx from different biological processes include pentose, phosphate shunt, citric acid cycle, or glycolytic pathway. The second factor is the hormonal factor. Alpha-adrenergic hormone, like epinephrine, inhibit insulin release, whereas beta-adrenergic hormone increase insulin release. Other hormones increase insulin secretion include chronic exposure to growth hormone, cortisol, placental lactogen, estrogen, and prostogen in pregnancy. The third factor is drugs. Sulfonylurea compound like glibenclamide, glazide, and glimepiride are used most frequently in the therapy of type 2 diabetes. Drugs that tolubutamide stimulate insulin secretion by the mechanism different from glucose. A new group of hypoglycemic drugs that is called the DPP-4 inhibitor, like sitagliptin, sacagliptin, and vildagliptin, stimulate pancreatic beta cell through increasing circulating level of endocrine that include GIP and GLP-1. Insulin metabolism. Insulin has no plasma carrier protein, thus its plasma half-life is very short. It's measured about 3 to 5 minutes. The major organ involved in insulin metabolism are liver, kidneys, and the placenta. Two enzyme systems are involved in its metabolism. The first include an insulin-specific protease, and the second is the hepatic glutathione insulin transhydrogenase, which reduce the disulfide bridge, resulting in insulin rapid degradation. Insulin action has got a major role in metabolism of carbohydrate, protein, and lipid metabolism. These include, first, effect on cell membrane transport. The first one is insulin, enhance the entry of glucose from extracellular to intracellular compartment using carrier-mediating facilitating diffusion in almost all body tissue, mainly adipose and muscle tissue, when the entry of glucose is followed by glucose phosphorylation and further metabolism. In liver tissue, insulin does not enhance facilitated diffusion, but it acts on enhancing phosphorylation of free glucose through inducing the activity of the enzyme, hepatic glucokinase, and in this way it enhances the glucose entry by simple diffusion based on concentration gradient of free glucose. Effect on glucose utilization. Insulin enhances the intracellular utilization of glucose, where about 50% of glucose is converted to energy through the glycolytic pathway, while the other 40% is converted to fat, and about 10% is converted to glycogen. It stimulated several key enzymes in glycolytic pathway, including glucokinase, phosphofructokinase, and pyruvate kinase. Insulin stimulates the conversion of glucose to glucose 6-phosphate by glucokinase, hexokinase 2 enzyme, mainly in the liver and the muscle tissue, which is then converted to glucose 1-phosphate that will be incorporated in the structure of glucose through the action of glycogen synthase enzyme, which is under direct stimulation by insulin. The effect of glucose on glucose production. Insulin inhibits the production of glucose from non-carbohydrate source, where it decreases the amount of key enzyme PEPCK, an effect that is usually opposed by glucagon, glucocorticosteroid, angiotensin, and other. The fourth effect is the effect on the lipid metabolism. Insulin is well known to have have a potent lipogenic activity. Meanwhile, it is a potent inhibitor of lipolysis in the liver and the adipose tissue. It inhibits hormone-sensitive lipase enzyme activity. It thus decreases the amount of circulating free fatty acid, and hence it affects the glucose metabolism as well as the free fatty acid inhibit glycolysis at many steps and stimulate gluconeogenesis. In patients with insulin deficiency, the hormone-sensitive lipase activity gets increased, resulting in increased concentration of circulating free fatty acid together with increased glucagon release, which oppose most action of insulin. The increased free fatty acid will convert to acetyl-CoA, which enter the citric acid cycle, convert to CO2 and water. However, in this way, or in this case, the capacity of this cycle is usually exceed, resulting in conversion of the excess acetyl-CoA into ketone body, which is called the acetone, including acetone, acetoacetate, and beta-hydroxybutyric acid in high concentration, resulting in ketoacidosis. The effect on protein metabolism, insulin is known to have anaerobic role in protein metabolism, where it enhances the entry of a neutral amino acid, into muscle cell, and thus a protein synthesis, and it, however, prevent protein. Glucagon is a single-chain polypeptide hormone, molecular weight nearly 3,500 Dalton, synthesized mainly in the A cell of the pancreatic islet. It consists of 29 amino acid, and it is also synthesized from proglucagon, a precursor molecule of nearly 9,000 Dalton. First, it shares some biological and biophysical property with enteroglucagon, which is released from the duodenal mucosal cell. It also flows freely in the plasma without being associated to any carrier protein, and thus its plasma half-life is only about 5 minutes. It is inactivated in the liver by removing the first 2 amino acids from the amino terminal end. Secretion of glucagon is inhibited by glucose, an action that emphasizes the opposing metabolic rule of glucagon and insulin. However, many other substances, including amino acid, fatty acid, ketones, GIT hormones, and neurotransmitter affect glucagon secretion. How glucagon action? In general, the action of glucagon opposes those of insulin, as insulin promotes energy storage by stimulating glucogenesis, lipogenesis, and protein synthesis. Glucagon causes rapid mobilization of the potential energy sources into glucose by stimulating glycogenolysis, and into fatty acid by stimulating lipolysis it is also the most potent gluconeogenic hormone. The liver is the primary site of action for glucagon, where it binds to specific cell surface receptor, and its action is mediated through cyclic AMP mechanism in the liver. It inhibits glycogen synthetase enzyme, and thus decrease glycogen synthesis, and meanwhile it promotes glycogenolysis. This action on glycogen metabolism is tissue specific, that is, it does not affect the muscle glycogen. It promotes the synthesis of more peptic enzyme, PEPCK enzyme, and thus the conversion of more amino acid to glucose through the process of gluconeogenesis, and this is the opposite action to insulin, which decreases the gene transcription of peptic PEPCK. In the adipose tissue, it stimulates the hormone sensitive lipase enzyme, result in increased rate of lipolysis. This enzyme induction is through increasing adipose cell cyclic AMP